now.
Okay, thanks everyone. Welcome to another week of the Behavior, Evolution, and Culture Speaker Series. We meet every week of the academic year at this time in this room, and we also simulcast on Zoom. So if you can't be here with us in person, then you can always join us on Zoom. Before I introduce today's speaker, I'm just going to give you a little bit of an overview of the rest of the quarter. We have two more talks, both of them in person. So next week, Sean Pearl is coming, and his talk is entitled Perceptions That Matter, Using Peer Evaluations and Dyadic Data to Explore Behavior in Rural Namibia. And the following week, Tom Kraft is coming from the University of Utah, and his talk is entitled Modeling the Dynamics of Infectious Disease Transmission in a Small-Scale Subsistence Population. So again, both of those will be here in person. So we look forward to seeing you for those. And you can see all the previous talks and future talks for next quarter all up on the website at bec.ucla.edu. I am very pleased to introduce today's speaker, Ainash Childebayeva, who is going to be talking to us today about population genetics and signatures of selection in early Neolithic European farmers. She's coming to us from the University of Kansas, so welcome.
Hello, everyone, and thank you for the introduction. I'm Ainash Childebayeva, and I'm really happy to talk to you about my research on the population genetics and signatures of selection using ancient DNA in early Neolithic farmers. But, sorry, I'm going to make sure that I can change this. When we think about selection, especially working in humans and like different examples of selection, some of the main ones that come to mind usually are high altitude adaptation in the Andes or Tibet, also the adaptation to being able to digest lactose into adulthood, also known as lactase persistence, adaptation to being able to live with infectious diseases, so pathogen adaptation and cold adaptation. But there's a lot more examples of that. And what's been happening recently in the field of genetics, but also generally evolutionary biology, is that for a lot of these instances of natural selection acting on humans, but also other animals and species, different genes and pathways have been identified that are under selection and have helped humans to adapt to different environmental conditions. And those are, for instance, if we think about like high altitude adaptation, like the work that Abby Bigham does here, which is the genes like EGLN1 or EPAS1. There's also genes that have been involved in lactase persistence, so that's MCM6, genes that are linked to skin color adaptation, so these are the SLC genes, SLC24A5 and 45A2. So there's many instances of natural selection operating on our genomes.
Can I just, interrupt you?
Yeah.
You have to unmute your...
Sorry. I was told to do that and I forgot. Now I'm muted, yeah. So, cool. And when we think about adaptation generally, I think when I think about it as operating on different timescales. So there are more immediate timescales that are there's like instances of physiological adaptation or homeostasis, and that would be, for instance, if you go to an environment that's really cold, and then your body starts shivering, and that's a response that is a more physiological response, that kind of this immediate adaptation. Then there's examples of developmental plasticity or developmental adaptation, and that can take place over years or decades. And there we think about usually like the early development having a big influence, because it is during early development that the human body is more plastic in a way, and that the environmental conditions during that time can have a big effect for later in life health and disease outcomes. And usually the big one that comes to mind is nutrition. So nutrition in early development is really important for the human body, and it can have big implications for later in life health and disease outcomes. So overnutrition or undernutrition, both. And there's also, of course, the genetic adaptation that takes place over thousands of years, like millennia, and the process that through which it operates usually is natural selection. So the selection of people with more fit to be in certain environments. So, and there are many methods that can be used to look at these different adaptive responses that operate over different timescales. And in my past work, I've looked at epigenetic methods and what we can learn from epigenetics when we're looking at adaptation, for instance, to high altitude. And today I want to talk to you about ancient DNA and what ancient DNA can tell us about genetic adaptation. So yeah, this will be the focus of the talk. And what has been great in the field of ancient DNA is that over the last 10, 20 years, many studies have come out that have published a lot of data on ancient DNA, and this is just a crude map of how many studies have been done with ancient DNA so far. I think this was the database was maybe from like 2021 to 2020, and you can see primarily the samples that have been published come from Europe because a lot of these research labs that do ancient DNA work are in Europe, but there's also a big variation in like how this work has been done. And I will focus primarily on European ancient DNA site, and I will talk about a study where new data was generated, but also a lot of the data that was used was from publicly available sources. And what's great about ancient DNA is that it can provide us with different insights into human past and natural selection that's operating on humans, because it really is creating this time depth to our ability to look at selection. And what's one of the great things about ancient DNA work so far was that we were able to track migrations and responses to natural selection through time. So like we all know that humans migrated out of Africa around, I don't know, 100 to 50,000 years ago and expanded throughout the world. And by studying populations from different time periods and comparing their genetic states overall in the population before and after a certain event took place, a cultural change or a certain environmental change took place, can tell us about what natural selection forces have been operating and what genes have been under selection and responded to that. What's interesting is that when we look at modern populations, we know that a lot of them are admixed, so they represent states of mixture of previously isolated populations. For instance, this is a very common plot that's used in genetic work. It's called the admixture plot, where individuals worldwide are compared with regards to their genetic structure, and individuals are painted using the genetic components that are defined by certain geographic regions. So in this case, we're looking at populations from Europe, America, and Africa, and more admixed population. For instance, when we focus on Brazil, we can see that individuals of Brazil from the study have primarily this orange ancestry component, but also they have some levels of the blue Native American admixture and the green African admixture. What ancient genomes can really allow us to do is to show natural selection in action. Like I said before, if we collect data from different time points from a population from a certain geographic location, we can track what happens with the allele frequencies before and after an environmental change and what genes and pathways are under selection. It also can allow us to distinguish between selection on standing variation and novel mutations. For instance, looking at modern data, it's really hard to do so, but if you have a certain gene that you're interested in, or a certain SNP that you're tracking through time, you can tell 10,000 years ago, was it there or was it not there? Maybe it's something that occurred de novo, or was it standing variation? And oftentimes, modern populations represent genetic states after natural selection took place, although it's still operating on our genomes. So by looking at ancient DNA, we can really get a better understanding of some of the selection coefficients that are interesting, like the selection coefficient itself, how strong the selection was it always strong? A lot of the time, the selection coefficient doesn't stay the same. We can also track when a certain selective pressure started operating on the genome. So we can track when it started and if there was any fluctuations in the strength of the selection. So I feel like that is a very powerful kind of viewpoint that is provided by ancient DNA. So for the study that I'll talk to you today, we will focus primarily on Western Eurasia and primarily in the Neolithic times. So I will talk about the adaptation to Neolithization in Western Eurasia. So the transition from a more, a transition from a more hunting and gathering lifestyle to a more sedentary agricultural lifestyle has happened multiple times in our evolutionary history, and it happened in multiple locations in the world. And one of the examples of that is the kind of the Neolithic transition in Anatolia, and then the migration of the farmers to Europe. And the Neolithic transition has been linked to multiple cultural shifts and cultural innovations. So it is during this time that we start seeing people living in houses. We see first domesticated animals, domesticated plants. First pottery starts appearing during that time, and also wells, for instance. It is also during that time that people start living in close proximity with each other and in a more sedentary way. So there's more of avenues for these diseases to spread between humans that usually, not usually, but oftentimes certain pathogens would jump from animals to humans. And then if humans are living sedentary and close to each other, there's a lot of kind of avenues for spreading the disease from one person to another. And one example of that is COVID. That's what's been happening for multiple years now. With regards to the Neolithic transition, what kind of has been hypothesized for some years before some of these genetic studies have come out, people weren't sure if it was the migration of humans from, for instance, I guess, sorry, this is not easy. It would be from here, from Anatolia and modern day Turkey. And what archaeologists have seen is that the actual method of agriculture has spread from first Anatolia to Europe, but for some time people didn't know if it was kind of a cultural diffusion or if it was a transition of humans. So yeah, people were debating, was it a diffusion of individuals or the endemic diffusion, or was it a cultural diffusion? So the motion of ideas, that was like the idea of farming that spread itself. Lately, I'm going to make this small. What has been shown using ancient DNA, for example, was that it was actually a migration of humans themselves. So if we look at the genetic structure of Europe through time, so if we start from 14,000 years ago to 6,000, not years ago, but BCE, what we see is primarily these individuals are hunter-gatherers. So they have this blue European hunter-gatherer component. They're called usually in this case, they would be the Western European hunter-gatherers. And some individuals here, more in the Balkans, so closer to Anatolia, have small levels of the Anatolian ancestry. So we start seeing it kind of shifting out of the Near East. Looking at the time transect from 6,000 to 5,500 BCE, there's greater levels of this Anatolian ancestry. So we see it starting spreading, kind of this Balkan route to Central Europe, where there's these higher levels of this orange Anatolian Neolithic component. But if you look at like Poland and then further to Spain, it hasn't reached it at that point by 5,500. But already by 5,000 BCE, this European, this Anatolian farming component is spread all around Europe with some admixture from the blue component, which is the hunter-gatherer component. And today I will talk about one of these examples of the early Neolithic farming community, and it's called, the culture itself is called linear band ceramic. It's from Central Europe. The name means linear pottery culture, and it is usually dated to between 5,500 to 4,400 BCE, or before Common Era. And these individuals occupied this area of Central Europe, and they lived in longhouses. So they were these houses that were occupied most likely by extended families who were farming land nearby and had some animals. And the name itself, linear band ceramic, stands for this pottery type that these individuals were making. The site that I will talk about is called Derenberg. It's from central Germany. It's also located close to other LBK sites such as Halberstadt and Mittelelbe-Saale. And the Derenberg site has been dated to 5,200 to 5,000 calibrated BCE. To give you some of the demographic context for these individuals, we generated data, genome-wide capture data for 32 new individuals. So that it was capture on 1.2 million SNPs across the genome. 18 were males, and 18 were females and 14 were males, and 22 adults and 10 subadults. And the individuals were dated to around 7,200 to 6,700 calibrated BP, which is before present. There was different laboratory and analytical steps were performed to be able to look at the data. So we took teeth as well as the petrous portion of the temporal bone. Then libraries were prepared, which usually contain both human DNA, pathogen DNA, if that person had a pathogen, also environmental DNA. So all kinds of DNA would be in a library. Then the library was shotgun sequenced to a shallow coverage, about 5 million, to determine if the sample had DNA. Then the samples that had high qualities of DNA were captured for human DNA and were sequenced again. After that, they were mapped to the reference sequence of the human genome. The DNA was authenticated to make sure that it was actually ancient, and for that, we look at these patterns of transitions at the ends of the reads. So because ancient DNA decays over time, what we see is that the cytosines get deaminated and they become uracils, but after sequencing, they become thymines after PCR. So usually at the ends of the reads that are more kind of open to it, we see this pattern of decreasing deamination of cytosine to thymine at the 5' end, and the 3' end for double-stranded libraries will have the opposite G to A, so guanine to adenine transition rate. So after we made sure that all these samples were clean, didn't have modern human contamination, and were actually ancient, we performed various genetic analyses such as PCA, admixture, and F statistics, and selection scans. So the first analysis that is usually done in the ancient DNA world, and also I think in the modern DNA world, is the principal component analysis, or the PCA. And here I'm showing PCA for West Eurasian populations. This is the scaffold of the PCA on which the ancient data is usually projected. So here the modern populations usually are distributed according to their geographic origins. So here in the top left are the Northern Europeans. These are the Southern Europeans here. These are the individuals from the Caucasus. And here is the cline of Northern Africa. And here now you see these modern populations are in gray, and the ancient populations are projected on top of the modern variation. And what you can see right away is that ancient populations are outside of the modern variation, which is kind of expected because, for instance, this group here are the Western hunter-gatherers. So these are the local hunter-gatherers that were living in Europe before the farmers migrated there. And these individuals are dated to more than 10,000 years ago. So they're quite far from the modern variation. These are the Eastern European hunter-gatherers. We're not going to be looking at them here, but the orange triangles here are the Neolithic farmers. They're closer to modern variation because in time also they're closer to modern humans, which also then makes sense that they will be closer to the gray points. When looking at the already published LBK data from other studies and plotting it on the PCA, what we see is that the LBK, or the linear band ceramic, fall kind of almost on top of the variation of the Anatolian Neolithic, but we see a slight shift towards the Western hunter-gatherers, so there's potentially this Western hunter-gatherer admixture. When we plot the new Derenberg individuals, who are these squares that are purple, they fall within the variation of the LBK and close to the Anatolian Neolithic, so we see that they're a typical LBK community with high levels of Anatolian ancestry and potentially small levels of hunter-gatherer admixture. After doing PCA, what we wanted to also do is to run admixture, which is also one of these hypothesis-free tests where all individuals are just compared to previous studies and the variation that we know already. So in this case, we compared them to already published individuals from different ancient studies. So here we see that the Derenberg cohort have high levels of this Anatolian ancestry, which is orange. So they're very close to the Anatolian Neolithic, but they do still have this blue Western hunter-gatherer component. So at like various degrees. So it's possible that they have this additional Western hunter-gatherer admixture from the local hunter-gatherers. And to test for this admixture component more formally, we used F statistics. F statistics are very commonly used in ancient DNA world, and the statistic that was used here is an F4. It's a comparison of 4 populations with one that is always an outgroup. In this case, the outgroup were the modern Mbuti. These are the individuals from sub-Saharan Africa, and we know that there shouldn't have any admixture or additional admixture, or there shouldn't be particularly closer to any of the other 3 populations. That's why they're used as an outgroups. And the other 3 are Anatolia, Neolithic, and P1, and then Darrenberg and hunter-gatherers. And what the test does is it just compares the number of the positions that are in this ABA state, which is has the P2 and P3 with the derived allele. They're sharing the derived allele here, which could mean a gene flow between them two. And the BABA state, in which case the derived alleles are shared between the P1 and P3. So that would mean more gene flow between P1 and P3. So these sites, the ABA sites and the BABA sites, are just counted, and then a difference between them is determined. So if for instance, the F4, when you subtract ABA from BABA sites and divide it by all sites, if that equation is 0, then there's no additional gene flow between these individuals. But if there's more ABA sites, so that show these derived allele at position 2 and 3, this could indicate gene flow between hunter-gatherers and Darrenberg. If it's opposite, so the positive F4, it would indicate gene flow between hunter-gatherers and Anatolia. So in this case, what we did was we compared, in this F4, Mbuti was our outgroup. We compared all of the published hunter-gatherer individuals from Western Eurasia, and we looked at Darrenberg versus Anatolian Neolithic. And again, if the F4 is negative, that indicates gene flow from hunter-gatherers to Darrenberg. If it's positive, it would be hunter-gatherers to Anatolia. And here, the F4 values are on the x-axis, and the hunter-gatherers are on the y-axis. And what you can see already is that most of the values are non-significant, so they're blue, but some of them are significant, which is shown in orange. And what we can see is that here on the bottom, negative part of the F4, we have a bunch of significant statistics, and these are mainly the individuals from the Western hunter-gatherer cluster. What we see if we go the other way, so the positive side of the F4 values, we see individuals from Anatolia Epipaleolithic, Israel Natufian, so this would be the gene flow from hunter-gatherers to Anatolia. And then we see populations that also make sense in this case. So what we did next is we compared different early Neolithic individuals from different studies from Europe to Darrenberg and looked at the levels of the additional hunter-gatherer admixture. And what we saw, I should have highlighted Darrenberg, but in almost all of the cases, Darrenberg is at the top of the list here. So they, compared to other Neolithic farmers of Europe, they have little amounts of this hunter-gatherer admixture. When you go into like middle Neolithic, which is on the bottom, the F4 values are very negative. So here in the middle Neolithic, we just see a lot more of the admixture with the hunter-gatherers. If in the beginning it's kind of low, towards the middle Neolithic, it really goes up. And what we did next is we compared the different hunter-gatherer groups to each other to determine which one is a better candidate for that. And the one that came up there is called the Loschbor. The hunter-gatherer individual is called Loschbor from the site where it's from, and we just determined that that was the closest hunter-gatherer source for modeling this hunter-gatherer admixture. Which was interesting was that we actually, in this test, some of these hunter-gatherers are actually from Germany, from very close to where Darrenberg's site is, and they did not come up as being significant. And Loschbor is, I think, in Switzerland. So, yeah. So then afterwards, when we saw that they do, in fact, have this additional hunter-gatherer admixture, what we did is perform QPAGM to actually test for the actual proportions of how much of this ancestry they have. And what we saw is that across the individuals here, they have between 0 and 10% of this Western hunter-gatherer ancestry, and the rest was the Anatolian ancestry in orange. Then we wanted to see when this admixture event took place. So we ran dates to look at when we can place this admixture of the Anatolian Neolithic and the Western hunter-gatherers. And according to the model, it was 25 generations ago, so about 680 years ago if we use like 28 years generation time. So most likely, as they migrated from Anatolia, the kind of admixture took place somewhere in the Balkans or something like that. After doing more general tests of population genetics, we wanted to see if any of the individuals in the group are related to each other. So we used pairwise mismatch rates. So looking at the mismatch, looking at the number of matches versus matches plus mismatches across individuals to see if we see any relatedness. So in this group, what we found was that there's 3 first-degree related pairs. So on this plot, on the x-axis are the different pairwise comparisons. On the y-axis is the pairwise mismatch rate. So the lower values on the y indicate more related because they have less mismatch. So none of them were twins. There's 3 pairs of first degree. There's a mother-daughter pair who were buried together. The daughter was actually a child. The mother was an adult. There was a mother-son pair. They were both adults. They were not buried together. And there was a sibling pair. We also found some second-degree related pairs, but most of the individuals were above second degree or unrelated. Looking at also the pairwise mismatch rate, we compared it by sex to see if there's any evidence for whether or not the males are more related to each other or the females are more related to each other at the site. And what we saw, if you compare male adults to female adults or male subadults to female subadults, there's no significant difference by sex. So we don't see any evidence of like a matrilineal or a patrilineal structure in this community, for instance. At least not looking at this group of 32 individuals. I think maybe with more individuals we can have a better sense of that. Afterwards, we wanted to look at the kind of a sense of what the population size was. So we ran an analysis of looking at the runs of homozygosity. Here in the Derenberg cohort, we saw that most of the individuals have very small ROH segments, which indicates a large population size and no evidence of inbreeding. Then we compared the German LBK groups to other early Neolithic farming communities from Austria, the Balkans, Hungary, and what we saw was that in the German LBK, the population size is large, or the ROH segments that we saw indicate a large population size, whereas, for instance, in the Balkans, there's more evidence of longer ROH segments, potentially indicating a small population size, or inbreeding, as well as in Hungary. So this could indicate a great increase in population size going throughout the migration. There was also no close biological relatedness between the LBK sites because some of them are very close to each other. For instance, Derenberg and Halberstadt are like 5 kilometers away and are more or less contemporaneous. So when comparing the relatedness of looking at all of the LBK sites across Germany, there was no close relatedness or less than third degree, which could indicate a big network of for this group of individuals maybe moving around a lot, or I don't know. So overall, like looking at the demography of the LBK, what we saw was that the Derenberg individuals in this case were very similar to other LBK groups in Germany with very limited additional hunter-gatherer ancestry. There's potential patrilocal structure. Actually, I don't believe that because none of it was significant. I don't know why it's still here. I thought I removed it. And we see a very low level of relatedness at the site, which could indicate a large population size for LBK. So after looking at a greater demography and comparing the Derenberg individuals to other LBK sites in central Germany, what we saw was that they're pretty similar and could be grouped in the same population to do these selection scans. And the question was, what are the consequences of Neolithization in this community? Because with the Neolithic transition, what happened was that the overall energy supply increased, going from a hunting and gathering lifestyle to a more sedentary agricultural lifestyle. We know that the population sizes went up and the living condition potentially got worse with higher exposure to infectious diseases, but also looking at things like bone markers of stress, which has been done for these LBK groups. They found more levels of protic hyperostosis and also other bone markers of them not having a really good diet in the early Neolithic. However, it becomes better in the middle Neolithic and later in the late Neolithic. And this could be also because of the risk of famine in agriculturalists, which could be higher than in hunter-gatherers. They're not agriculturalists, but they're also more susceptible in the sense because they are eating a more energetically rich but a less diverse food source, and they're more susceptible to harvest failures and pests. So we can see that there are multiple ways in which they're experiencing these environmental pressures for a natural selection to operate. So various two types of selection tests were performed in this group, the frequency-based and haplotype-based. So a frequency-based test was a comparison between populations, but for the haplotype test, the individuals were also imputed, but also compared to different populations. So the first test was called LSBL, or locus-specific branch length, and it's calculated based on pairwise FSD. What it does, it isolates changes on a lineage of one population compared to two kind of outgroup population by of looking at the levels of the branches, at how long the branches are based on the pairwise FSD. In this case, we compared the LBK farmers, so in green, to individuals of the Anatolian Neolithic in orange, and to the various Western hunter-gatherers in blue. After performing this test for 1.2 million SNPs across the genome and comparing them to each other, we focused on the 1% top hits. What we found was that looking at the pathways and the SNPs that were found in this top 1%, there are two pathways that came up as being significant consistently, and that was the immune system and the metabolic system. And the immune system, within the immune system, it was genes in the MHC, some HLA SNPs, but also gene HNF alpha. And the pathways that came up as being significant were phagocytosis, immune system process, leukocyte migration, and homeostasis. In the metabolic system, some of the genes that were interesting were the FAT2, which was found by other studies as well. Then this leptin receptor gene, the pathways were the regulation of triglyceride lipase activity, lipoprotein lipase activity, and fatty acid transport. And this LEPR receptor was interesting to me also because it is at the intersection of the two pathways, both the immune and the metabolic, which could indicate, but it also makes sense that these two pathways are related to each other. They don't exist in isolation. There's a lot of overlap, and the nutrition has a big effect on the immune response. Afterwards, we designed our own test to look at selection here, which is called AIMLess. It stands for Admixture Informed Maximum Likelihood Estimation for Selection Scans. And what's different about it is that it is able to test for signatures of selection in an ancestry-aware fashion, which is different from other tests, and it takes into account the admixture proportion. So in this case, performing a lot of these population genetic analyses beforehand was very important to us because we found that these individuals could be described as primarily Anatolian Neolithic with some Western hunter-gatherer ancestry. And what this test does is it finds significant frequency deviations that are for SNPs that are deviating from this admixture ratio that was found. And based on the AIMLess results, here is a Manhattan plot across the 22 chromosomes, and the p-values are on the y-axis. And what we saw was that the HLA pathway came up as being significant, which was not surprising, but also some other genes that are involved in both the immune system and the metabolic system. Afterwards, we wanted to do some haplotype-based tests. So for that, we used GLIMS for imputation and phasing of these individuals because we actually had pretty good preservation for the samples. A lot of them had enough SNPs to be imputed, and the imputation briefly is a procedure of kind of filling in the blanks where we, for instance, for SNP data, we get just the variation at certain loci, but then we can match it to a reference database of haplotypes and then fill in the blanks using the reference. In this case, we used a modern reference, which is, of course, a limitation for ancient DNA studies because we're kind of filling in based on the modern variation, and we know that maybe some of the variation that existed in the past is not here anymore, but it's anyway, it's kind of something that we have to deal with, and so I usually, these haplotype-based tests after imputation, I usually take it with a grain of salt. However, before doing any of these tests, the samples were tested, or we performed some testing of the imputation. So in this case, what we did was we used a 99x covered sample that was actually a positive control in all of our analysis. So it was like sequenced many times, and we merged everything, and we downsampled to 0.05x to from 0.05x to 4.5x in coverage. And I think at each level, there were 10 different downsampling experiments, and then we imputed at every level. And what we see here is that the mean coverage is on the x-axis, and on the y-axis, we see the kind of the diploid call agreement of the various imputed samples to the 99x covered individual, and at 0.5x already we see 98% agreement. So that was kind of our cutoff for sample inclusion in this case. And by 0.5x, I mean if it's a sample with 1.2 million SNPs, then before imputation, it would have more than 600,000 for our inclusion. So that was our sample inclusion threshold. However, I think there's a lot of caveats to imputation with ancient DNA. For instance, a downsampling is not a very good experiment to what actual low coverage sample looks like, because downsampling usually is more homogeneous, while ancient DNA samples are not homogeneous in their coverage. Some regions may be well covered, some regions may be poorly covered. So there's a lot of caveats to consider in this case. But after doing all these experiments of imputation and actually imputing the data, we performed what is called the extended haplotype homozygosity test, known as XBEHH. And what it does is it can identify selective sweeps across the genome. And in this case, it measures the amount of extended haplotype homozygosity for a given SNP along the genome, and it compares the ancestral allele versus the derived, in this case, haplotype. So, for instance, it would be if, for instance, this is a position on a certain chromosome, and we have the extended haplotype homozygosity level. In this case, the blue is our ancestral haplotype, and the red is the derived. So here, what you can see is the derived haplotype is longer than this ancestral, which is kind of this just peak. And in our case, the ancestral would be the hunter-gatherer haplotype, and the derived would be the farmer haplotype, for instance, or something like this. And we were comparing the farmer population and the hunter-gatherer population, and this test was done across the genome. So some of the loci that were identified were already known as being under selection, and it was good to actually find these loci also and to confirm these analyses. So one of them was a skin color associated SNP in SLC 24A5. We know that it's under selection in Western Eurasia. And what we saw was that, for instance, if you looking at the hunter-gatherers, and here the different haplotypes are shown in different colors. So what we see is that the derived haplotype here is not present at a higher level, although it is present in some hunter-gatherers, actually, for instance, the Eastern European hunter-gatherers. But when we look at the early farmers, what we see is that primarily they have the derived haplotype compared to the ancestral, which could indicate that this SNP has been under selection in this population. Another locus that we identified is linked to a metabolic adaptation, and it's a SNP in FADS1. It's a haplotype in FADS1 that is associated with the ability to digest fatty acids. And the hypothesis was that it is under selection because of the moving from the animal fatty acids to the plant fatty acids, that people potentially weren't as well adapted to digesting, and that's why it was under selection. But what we see in hunter-gatherers is that they primarily have the ancestral haplotype here in blue. So there's actually some potentially purifying selection happening in the hunter-gatherers where we don't actually see the derived haplotype at all. And then later on in the early farmers, there's the relaxation of this selective pressure, and we start seeing more of the derived haplotype in the farmers, but also some levels of the ancestral as well. We also identified some new signals of adaptation comparing hunter-gatherers to the early farmers, and one of them is this LMF1 gene, and it is potentially under selection in the early farmers where we see these the derived haplotype. We see a longer derived haplotype comparing to the hunter-gatherers. And another one, which was also in the immune function pathway called JAK1, so that there we also see potential selection in the early farmers. So that we see that these pathways, the immune and the metabolic pathway, are showing the evidence of selection. And I think it also makes sense for this specific process, so the Neolithic transition, because the pathways in a way are very linked. So for instance, in times of optimal nutrition, what we see usually is a normal immune function, but in times of malnutrition, for instance, we could see potential immunosuppression and higher susceptibility to infection. But in times of overnutrition, which is something that maybe is more present currently, is we see immunoactivation and higher susceptibility to inflammatory diseases. So in summary, based on our selection scans, we saw evidence of selection in genes that are linked to metabolism, to immune function, also potentially skin color, and it could indicate this ongoing adaptation that's happening in the two neolithizations through this combination of immune and the metabolic systems. And I think I have a bit of time, and I just want to talk a little bit about some of the ongoing work that I'm doing. And in this case, it's a different part of the world, and it is Central Asia. It's where I'm from, actually. And I'm also interested in doing some population genetic work and looking at selection in the Central Asian population. And what's interesting, or actually people now want to call it Central Eurasia, not Central Asia, because it is kind of in the center of the Eurasian continent. And we know that it has experienced multiple waves of human migration, but also hominin migration. For instance, some of the very famous sites, like the Denisova cave, it's in the Altai, but it is very close to Central Asia. So there we find evidence of Neanderthals, but also Denisovan individuals. And also through time, there have been multiple waves of migration through the region. And what I'm interested in, so currently what I've been working on is collecting samples from Kazakhstan, Kyrgyzstan, and Russia. So this would be Kazakhstan, this is Kyrgyzstan, and then this would be Russia. So creating a database of multiple individuals to then perform whole genome sequencing. So I'm interested in generating high coverage genomes to then perform various more detailed tests of demography that is not possible to do with genome-wide data. So I'm interested in looking at archaic introgression, so looking at how the introgression from Neanderthals and Denisovans, how this introgression changes through time in Central Asians, for instance, to see if there are any loci that are potentially adaptive, because recently studies have shown that introgression has been linked to selection. For instance, in EPAS1 for high altitude adaptation, it's also been shown to be linked to risk alleles, such as with COVID. So I'm interested to look at kind of the architecture of the introgression to see where are these loci that have more introgressed elements, what are the regions that don't have introgression. Maybe this can tell us something about the population history of this group. I'm also interested in doing some selection scans on whole genome data to, again, like looking at haplotypes and how they change through time, and to connect it to various phenotypic outcomes and health and disease outcomes. And yeah, so some of the ongoing research questions are looking at the nature and chromosomal distribution of introgression from Neanderthals and Denisovans, and for that, really, this high coverage genomes would be very helpful, because a lot of the capture data sets don't capture the archaic introgression. I'm also interested in looking at population sizes and times of admixture of the ancestral populations that form the Eurasian genetic pool, and also I would like to do some genome-wide scans of selection to identify candidates of natural selection acting in the Central Eurasian population through time. And with that, I'd like to thank everybody who made this work possible, and thank you for your attention.
time for questions.
Hi, really cool. I was curious because you have your aimless scan and also the XBEHH, and it looks like a lot of the selection seems to be on standing genetic variation, particularly from like the Anatolian farmers. That's my correct understanding. So do you tend to see that aimless and XBEHH sort of produce the same signatures of selection, the same targets of selection, or are there differences? And if so, why might that be?
I think there are both similarities and differences. I think the frequency-based tests comparison with the haplotype-based tests are just looking at different time depths, I guess. With the haplotype-based tests, we would see more selective sweeps and the more recent potentially selection, and then comparing like frequency-based tests, comparing different populations, you get at a more ancient selective, I guess, pressures that have existed since the split. But yeah. But yeah, so it's like both. It's overlap, and there's also some, there's, we did look at the sites that are overlapping between, LSBL, XBHH, and aimless. So, yeah.
Would you mind when you get the questions just repeating them or summarizing, just because the Zoom participants can't hear always what the audience is saying?
Yeah.
Thanks so much. Oh, and there's a question in the chat. I'll read it to you. Thank you, very interesting. In these types of analyses, like extended haplotype homozygosity, is there a way of distinguishing a selective sweep from violent encounters between groups? I see a risk of confounding these events, natural selection versus group dominance, if the dominant group has low heterozygosity, or would the selective sweep signal still be apparent regardless?
That's an interesting question. I'm not sure. I have to think about how do you compare a violent encounter versus a selection sweep? I guess in that case, it would be like if one population comes in and completely wipes out a different population, so it's their haplotypes that are becoming more present. I mean, and then you compare two populations with that. But I mean, you do this analysis genome-wide, so like you don't look at just one locus. So I think genome-wide, you would still, if it's a violent encounter and like a completely new population is replacing another population, then genome-wide it should look kind of similar. But then if it's a selection event, there would be peaks that are different, I think. I don't know. What did you think of? Yeah, I think to me that would be, yeah.
You'd also have to have the same, if you had two populations coming together, regardless if it was violent or not violent, if it was replacement, if you saw a selection scan and it was indicating selection at a locus, if you saw that extended haplotype homozygosity, that would mean that dominant, the group that replaced would have to have had this selective event.
Exactly, yeah.
Right?
Yeah.
Yeah.
Yeah. And it was operating in the group that replaced. Although, I mean, in this context, we actually don't know what kind of event was the replacement of the, not replacement, I don't like the word, but like the migration of farmers to Europe, because there's very little evidence on how the actual interaction between the hunter-gatherers and farmers was. We do see some evidence of violence between the different hunter-gatherer groups, but the nature of the interaction with the farmers and the hunter-gatherers, I think, is unclear. What we do see is that the level of mixing goes up through time. So it seems like in the early Neolithic, they're not mixing. Later on, they're starting to mix more. So the level of admixture goes up to 20-30%. And now, if you look at humans currently, I think in Europe, there's maybe 20 or 30% of this hunter-gatherer ancestry still present. So, yeah, I don't know. Yeah.
I have a couple of just small questions about the demographic part of the talk. This was all really interesting. So, the part where you were talking about sex differences, and then you made an allusion to that you couldn't determine whether they were matrilineal or patrilineal. One thing I was thinking is that I know you said you had quite a few children in the sample, right? And so I think you would, so you need to account for that in a way to figure out matrilineality,
We did that.
right?
Yeah.
So you excluded the children
Yeah, I can show.
and then saw no difference?
Yeah, I went through it a bit too fast. Yeah, so here the 4 lines are, the red is female adults, the orange is female subadults, and then the blue is male adults, and the light blue is the male subadults.
And the y-axis is, so higher on the y-axis means more or
Less.
people that, higher on the y-axis is fewer people that you're related to?
Yeah, so the males are lower, but you know, the when we did statistical analyses, this was not significant.
Yeah, that's really
Yeah.
yeah.
Yeah. But it could, I don't know, I think in the early Neolithic, we don't know much about the population structure. We know that towards middle Neolithic, already you start seeing these, you know, patrilines, where if you look at 5 generations at the same site, they will have like the same Y haplogroup, and they would be related through the paternal line. So it starts to, I think, develop later. Yeah.
Because this looks more like a sort of multi-local hunter-gatherer kind of.
Yeah, it seems like there's a big network, and potentially also still interacting with Anatolia, because the population size at that point is huge. So there's potentially, I think, the theory is that as they're migrating out, and they're probably leaving Anatolia because they're running out of land, and as they're going through the Balkans, and then Central Europe is, at the time, I think it was pretty arable, and there's a big kind of landmass, so then they could really increase the population size.
And I have one more sort of quick question. So when you were, I think it was like the next couple slides, you were talking about whether there was sort of potential intermarriage between groups
Yeah.
and things like that. But I was just like, to get the degree of relatedness where you would have close relatives in the other group, that temporal gap would have to be pretty tight, right?
Yeah,
Because otherwise it would just be like second cousins or something,
Yeah.
right? And I wasn't sure how accurate the dating of any particular individuals are, so that like can you rule out intermarriage or?
I mean, it's kind of hard, I think, and we didn't date everybody, right? In this case, only about 10 individuals were dated, and there's a range of how accurate these dates are, so the radiocarbon dates. So,
What's the range like? Is it like 5 years or 5?
I mean, in this case, for instance, it's like 100, 200 years.
Yeah, okay,
So
so
that's-
it's big enough that you probably
Yeah.
couldn't.
So, but then we looked at like less than like 3rd degree, so that's still not so much. But I think what would be great to do, and I haven't done it yet, is what people are doing now is they're looking at IBD. So sharing, where you know you just impute and then you look at IBD, and then you know still you're using imputation, so that's not ideal, but that's currently where I guess the field has moved a lot into that direction, and it does work. I've seen, you know, where people you can find up to 5th degree related individuals, and it is very interesting looking at like relationships between sites. And then it's actually possible, because with the frequency-based methods, you cannot differentiate beyond 2nd, 3rd degree. It becomes all kind of similar, and it's because you know the coverage and stuff like that. Yeah.
Ainash, I have a question sort of picking up on the demographic vein. Thinking about sex-specific admixture patterns, so when you have a little bit of hunter-gatherer ancestry, right, in the Berenberg sample, have you looked to see if that is coming from one direction versus the other?
Yeah, actually, that's
Male
a great
versus
question.
female?
Yeah. We haven't looked at like males versus females.
Okay.
It would be cool. Yeah, we could also look at the
X
Yeah.
chromosome data, right?
Because like X chromosome, they do have both. Looking at Y haplotypes, they do have both farming and hunter-gatherer Y haplotypes in the sample. So it seems like and the maternal also it's both the mitochondrial. So, but yeah, I agree. It would be interesting to look at like X versus
But you do see,
Yeah.
both if you look at mtDNA and Y.
Yeah.
Equally.
I don't know if it was equal.
Maybe
I don't
not.
remember. But I just remember seeing both.
They were both.
Yeah.
I have a couple of questions. One actually is about this slide. So these are these individuals, are they a bit older than a few slides before when you were showing the genetic variation very dramatically changing from hunter-gatherer to the
Yeah.
the more like farmer, right? Aren't these a bit older? So how does that compare within
So,
that
The question
transition?
was, are these individuals on this slide older than the ones from here? Yeah, and so here they would be, so LBK is like 5,500 to 4,400, so they would be like here.
Okay, so it
Yeah.
was just the difference of the BP versus the
Yeah, yeah.
Okay.
Sorry, that's that's BP versus BCE. Yeah. So by this point, Germany is of course that we see that covered here, but we also see this orange ancestry here in Germany as well.
Okay.
Yeah. But not so much further out. Also in the UK, for instance, by that time they don't get there, or like Sweden, like Scandinavia. It's just kind of this like diffusion of people. So. Yeah.
And then my other question is about your upcoming work, Kazakhstan and Kyrgyzstan. So are you looking at this time period in that area, or are you looking at a different time period? Are you like interested in a particular cultural transition, or just more broadly at transition in the region?
So the question was about the work and ongoing work. So I'm interested in the change through time. So a lot of it is also going to be using published data, because it's just it's out there, and I think that's a great. What I like about the field is that a lot of the data is made available, the genomes. So I will be incorporating a lot of the published data, but also producing data as well. So like high coverage genome data. I mean, it's very difficult with ancient DNA in the sense that there are so few samples that have enough coverage, enough endogenous DNA to begin with for it to be economical to then create a 30x covered genome. Because
What's the typical coverage? Is it 0.5?
no, typical coverage. So the typical coverage would be, it varies by geography, and it varies a lot. For instance, in the Caribbean, you have really bad coverage usually, or like if it's a sample that's coming from like the jungle, your coverage is not going to be really great because of preservation. But samples from like Europe, samples from Kazakhstan usually are okay because the climate is a bit drier. So usually for I guess, I don't know, it really varies the coverage, but usually we don't see anything that's like very high. You see around like, I don't know, from 0.1% to like 5%, 20%, and then you just go from there, and then yeah. More questions for
So you didn't find lactase
No, lactase happens later.
later. And so the LBK pottery, were there any like residue analyses to look at?
I don't know about that. Yeah, that's a great question. Maybe, I don't know about that, but lactase, I think, based on recent data, starts in the Bronze Age. So it's coming from, pastoralists who are coming to Europe from Eurasia. Yeah.
I was going to ask relatedly, is there any evidence of, I know you mentioned agriculture, but of animal husbandry?
Um,
in this population?
There is evidence of animal husbandry. I'm not familiar with it as much, but yeah, they did start domesticating animals during the Neolithic. So I don't know if they migrated with the animals. It's possible. I think they were using animals. I'm not sure which ones exactly, but yeah. Yeah.
Hi. Just curious, in your upcoming research, are there any biological pathways that you're hoping to pick up on, or you think you might find in your selection scans in Kazakhstan?
Well, also, I think metabolic pathway I'm thinking about, because right now we see higher levels of metabolic disease in the population, and I'm curious to see if that's like a recent thing or if that's been around. We also see high levels of cardiovascular disease, so I want to look at these pathways. Maybe there's something that's in the population history that at some point maybe those pathways were positive, and then currently with the current lifestyle it was flipped, as it somehow happens sometimes with the modern lifestyle and diet. But yeah, those are the two that I'm interested in primarily. I mean, there's other pathways, of course, like the pathogen pathways, because of the different, and that would be collaborating with people who do pathogen work, so to see what pathogens have been around in the area, to see then if there's any pathogen selection happening. Although there also, I guess the difference then, if we have a more nomadic population, and there's also some parts that are more sedentary, so the nomadic populations would be potentially less exposed to pathogens, whereas the more sedentary populations would be more exposed. So I think in that sense, it could be interesting as well. Yeah. Obed?
With respect to your aimless analysis, I was wondering how exactly it was that you incorporate the other ancestries into detecting
Yeah.
selection.
So the aimless analysis, it has when it looks at the mixture, it has the priors. So the priors are the ancestry proportions from other analyses. In this case, we just used like 95 and 5, and then whatever was deviating from that was considered potentially under selection. Of like deviating from the mixture, I guess. Yeah. What?
Kind of like a delta ancestry scan where you look at.
I'm not familiar with the delta ancestry scan.
Oh, it's like it's basically you take the genome wide, you divide it up, and then based on the mixtures, you look at the deviations of the proportions of.
Yeah, because if you expect, I guess genome-wide we expect a certain proportion, but then looking at the different loci to see like which loci really deviate from that. Yeah. Yeah.
For this particular population, have you been able to look at introgression?
No, I haven't looked at introgression because of the nature of the data. Of the capture that was used here is not informative of introgress sites. In this case, we would need whole genome sequencing data.
Even with the post imputation?
Yeah.
Okay.
Not so much because we use 1,000 genomes for imputation. I don't know to what degree 1,000 genome SNPs are informative for introgression. Yeah, because I don't know. But then I think it would be primarily then focusing on whole genome. Although we did generate 2 whole genomes and they were published as a part of the study, but I haven't done this analysis. Also, introgression at that point shouldn't change so much, I think. If the introgression happened 40,000 years ago, or like 50,000 years ago, they should all have similar, not similar, but 1 to 2% introgressed elements in the hunter-gatherers and the farmers, all the Eurasian populations. But it would be interesting to see how it varies by population, I guess.
I think it'd be interesting to see how it varies in this turnover period as well, too. If something was potentially more beneficial or deleterious.
Yeah, well, someone should do this work. The data is out there, yeah. Yeah.
Great, thank you so much.